And it is my great pleasure to introduce Tori Fox and to give the floor to her.
Great, thank you, John. Thank you for that lovely introduction. It truly has been a long path here at the University of Washington, and I am very excited to finally get to share all the research that I have been doing these past 2 years with all of you. So, to begin, I want to introduce my thesis research, Understanding the Microbiome of Puget Prairie Plants. For those who don't know, Puget Prairie ecosystems are prairies that occur throughout the Pacific Northwest. These prairies notably occur in Washington, Oregon, and British Columbia. These prairie ecosystems are rich in species diversity with over 190 different species of plants. These Puget Prairie ecosystems shelter threatened and endangered species of plants and animals. These are just a few of the Puget Prairie species that I chose to look at. My species pool was 16 species large, and I wanted to show you these images just to show you the sheer diversity of shapes and colors and sizes that the Puget Prairie plants come in. Also, throughout the presentation, I'm going to refer to these species using their species codes because their species scientific names are quite long, and it would be a bit of a burden to have to say it every time I referred to each species. One of these species that I would like to highlight is Castanea levisecta, which is the golden paintbrush plant. The golden paintbrush plant is a threatened prairie plant species which occurs throughout Washington. This plant is a hemiparasitic plant, which means that it has root connections from its roots to nearby host plant roots. It uses these root connections to absorb nutrients, plant defensive compounds, and other building blocks for life from its host plants. Our lab group is interested in knowing if these root connections may also provide a pathway for bacteria to travel between plants in the Puget Prairie ecosystem. We're interested in looking at the Puget Prairie microbiome because not much is known about the bacteria that inhabit these Puget Prairie plant species. No one has performed an in-depth survey of what bacteria exist in the Puget Prairie plants. So that is what I decided to base my research off of. I chose to examine the bacterial microbiome of just 16 of the different Puget Prairie plant species. As I was performing this research, I came up with two purposes. First, what bacteria exist within the stems of prairie plants? And second, does plant parasitism help bacteria travel between plants? As I was investigating these Puget Prairie ecosystems, we chose two sampling locations. First of was the Smith Prairie, which is on Whidbey Island in Washington, and the second, which is Glacial Heritage Preserve, which is just south of Olympia in Washington. At Glacial Heritage Preserve, we were interested in studying research plots that had received different treatment types. These treatments were applied at Glacial Heritage Preserve for several years on different schedules, where they used a combination of burning and mowing techniques on these different plots, where plots were either burned or mowed once a year or burned once every 3 years. And I'll be going into this part of my study in more depth later on in chapter 1. In order to look at the bacterial microbiome, first I had to collect my plant samples. In order to look at their microbiomes, I sampled plant stem samples, and as I was sampling these stems, I sterilized them in the field, sterilized the exterior of the stem, in order to just look at the bacteria that lived inside of the stem itself. I sterilized the stems and the equipment I used to collect the plants in order to remove all the external bacteria from these stems so that they wouldn't contaminate my samples. I intentionally chose to sample stems only from healthy plants, from plant species that were found frequently enough throughout the prairie so that I would have a large enough sample size to use to compare between plants, and I collected from plant trios. So this is sort of a demonstration of what a trio might look like. So remember, we have our parasitic Castanea levisecta plant here. I collected potential host plants that were found within 4 inches of these Castanea plants. We were not able to confirm parasitism in the field because in order to do so, you would have to dig up the Castanea and look at its roots and find hostorial root connections, which are little organs that connect the host and the parasite roots. And we didn't want to do that because digging up the Castanea, of course, would hurt this threatened plant species. So, because Castanea roots are not very long, we decided to collect plants that were only found within 4 inches in order to assume that that was acting as a host plant for our Castanea. In order to assemble the trio, I looked around and tried to find a plant of the same species as the host that was at least 2 feet away from our parasite and our host, but within either the same plot or a nearby plot as the other plants. I wanted to use this non-host to compare to the host plant, and I collected it from nearby in order to make sure that differences in microclimate wouldn't have too much of an effect on the bacterial microbiome of the stems that I was collecting. And of course, I will get into this at the very end of my presentation as I dig into chapter 2. In order to look at the plant microbiome, I took my stem samples and ground them into a fine powder using a mortar and pestle and liquid nitrogen. From this powder, I extracted bacterial DNA using a QIAGEN DNA extraction kit. Once I had extracted my DNA, I sent these extracts to the University of Minnesota Genomics Center for sequencing. We sequenced the 16S gene because this gene is often used to look at the taxonomy of bacteria that we have and can sometimes be used to identify down to the family or genus level of the bacteria. Once the University of Minnesota Genomics Center was finished with sequencing, they sent me back a series of raw reads, which I processed using CLC Workbench software. So many of you are probably fairly unfamiliar with what DNA sequencing is like. So raw reads that I mentioned before are basically strings of nucleotides, which are comprised of A's, G's, T's, and C's. These reads are sequenced by Illumina technology and must be processed before we can extract any useful information from them. So once we're given the raw reads, I trim them and filter them for quality. Then, once these reads are processed, I bin them into OTUs, which are operational taxonomic units, where similar strings are all binned together and dissimilar strings are put into different bins. OTUs, you can sort of think of them as like a unit of taxonomy. You can't always use DNA to identify down to the species level, but you can use DNA to examine the different levels of taxa that you have. So that's a very complex idea, and I want to break that down into a bit more of an understandable way. So for example, let's take 3 different nucleotide strings. These are our raw reads, which have many base pairs coming off to the sides. This is only the middle part. Because the ends of strings are usually low quality, we remove the ends. So this is the trimming process where we cut those ends off. Then we take those reads and we look at the actual content of the reads themselves. So each base pair gets a quality score assigned to it, and strings that have low quality overall are filtered out of the process. This can also include strings that are a little bit too short or are much shorter than our target string length. So then, after we have filtered and trimmed our reads, we bin them into OTUs. So remember, reads that are similar are binned into the same OTU, and reads that are different are binned into different OTUs. In this case, our 2 strings are quite different from each other, so they are binned into 2 different OTUs. It's incredible the number of reads that are present in each sample, which can range up to thousands of reads. Our reads, once we have our OTUs assigned, we then compare them to a database of known bacterial DNA to find a close match. If these reads are within 97% similarity of a string that we know, we can identify the taxonomy that that read represents. So you can think of it sort of like a criminal database. If you have a thumbprint and you upload that and compare that to a database, it can tell you who that thumbprint might belong to. However, sometimes we don't have enough information, and the reads that cannot be identified, if you upload your thumbprint to the database, it flashes an error and says that has not been uploaded to the database yet. We don't have an identity for that read. We call those kinds of reads de novo reads. To make sure that I was only working with quality samples, I made sure to collect many more samples than I could actually process. So I collected 387 samples, but I only processed 335 of them. We also had budgetary constraints, which limited the number of samples that we could process, because sequencing, although it's gotten a lot cheaper in the past few decades, it's still quite expensive. After I had processed my samples and done a bit of pre-analysis, we discovered that the samples processed in batches 1 and 2, my two earliest out of 13 total batches, showed evidence of contamination that I had to remove. Or at least evidence of contamination, which meant that I couldn't use any of those samples present in batches 1 and 2, which were about 42 samples. Samples 55, 258, 438, and 439 were outliers, so we also removed them from the data set. And that left me with 292 quality plant stems to examine. I performed data analysis on these plant samples using our coding software, which is software commonly used by ecologists. So remember back to my research questions. I decided to break down my first research question into 3 different parts. Does bacterial OTU composition of samples differ between plant species, differ between sampling location, and differ between disturbance regime treatments? And second, does the bacterial OTU composition of hosts and are the bacterial OTU compositions of host plants and parasites, remember our Castanea and the plant that it's parasitizing, are those bacterial OTU compositions more similar than the non-hosts and the parasites? So I would like to share with you my results. So overall, I found that there were 7,365 total OTUs contained across all of my plant samples. So there's just a sheer, there's a huge amount of bacterial diversity that's present in these plant samples. Interestingly, 3,375 OTUs, nearly half of them were completely de novo bacteria. So there is still so much to learn about these microbial systems. We just don't have the capacity in the database yet to even understand what nearly half of my bacteria are. Now I would like to point you towards this figure. We have number of OTUs on this side and plant species along this axis, where each data point is one plant stem sample. So you can see that there's a huge range in the number of bacterial OTUs present in my samples. So take this Acme, for example, there were 600 bacterial OTUs in this sample, and compared to Okfo, we only have about 50 bacterial OTUs in this sample. So we don't really know yet what's driving these differences in the number of bacterial OTUs that are present in each sample, but that's something that we would like to investigate in the future. Next, I want to talk about what kinds of bacteria were present in my samples. So in this figure, instead of looking at the number of bacterial OTUs I have read abundance. And this just sort of shows us how frequently the reads are shown, because you can have many, many different reads comprising one bacterial OTU, and sometimes OTUs are only comprised of a few different reads. So that doesn't really show us the proportion of the bacteria that may be represented in my samples. So if you look across, you can see that 5 different bacteria were dominating my plant samples. First is actinobacteria, which is represented in this first part of the bar graph. Next is Bacteroidetes, which is represented in a smaller proportion across most species, the second bar in the graph. The third is Cyanobacteria, which for the most part largely dominates most of my plant samples. That was interesting to note because it didn't seem that any other bacterial phyla were that frequently observed in my samples. Firmicutes was also present in some samples, but in a very small proportion for most plants. And second was Proteobacteria, second only to the Cyanobacteria that was present in my other samples. So that is interesting information for us to note, because now we can examine not only how many bacteria we might have in our samples, but also what kinds of bacteria might be represented in these plants. So now I want to address my first question: differences in bacterial OTU composition between plant species. So now I'm going to talk a bit about bacterial OTU compositions. It's a long word, but essentially I'm asking what makes up the microbiome of these plants. So in this stem, for example, let's compare the stems of Urla and Demi. This is a picture of what bacteria live inside those stems. The way that we look at composition is basically a factor of frequency, how often a bacteria exists in a plant stem, and presence, if a bacteria even is present in a stem sample or not. So, for example, we may have bacteria that are shared between some OTUs but in different numbers. So we have one bacterial taxa that's present in Urla once, and we have that same bacterial taxa multiple times in Demi. And with this green one, it's sort of the same but reverse, where we have one bacterial taxa that's present frequently in Urla, and that same bacterial taxa that's only once in Demi. Then we have our composition that's partly determined by presence of bacterial taxa. So Urla is the only plant that has this bacterial taxa, the triangle, and Demi is the only plant that has this bacterial taxa, the oval. So overall, these plants, you would say, have different combinations and composition of bacterial OTUs. So in order to look at my plant species, I performed a Permanova test to see if these plant stems have a difference in OTU composition based on species. So I had a fairly large sample size to work with, my 292 samples, and I set my alpha value to alpha equals 0.05. And that's basically statistics talk for if my p-value is larger than 0.05, there are no differences. But if my p-value is less than 0.05, then there are differences in whatever factor you're looking at. In this Permanova test, I found that my p-value is 0.01, which means that there are some very large statistical differences in OTU composition based on species. So I performed a follow-up pairwise test after my Permanova test, because the Permanova doesn't tell you between which species there are differences. The pairwise test pits plant species against each other to examine if there are differences in their bacterial OTU compositions. So let's take Acme and Akvo for an example. This is the p-value between those two plants, and we can see that the p-value is very, very small, where it is much less than 0.05 at 0.01, And that means that there are serious differences in their bacterial OTU compositions. So those plants are different from one another. And you can see throughout the rest of my data that almost every other box is green. So that indicates that plants usually have seriously large statistical differences in their bacterial OTU composition. However, there are some red boxes throughout my data. And this indicates that plants actually have similar OTU compositions. So I wanted to understand why there were some similarities between these plants. Looking at this example, this is loader and lout, and both of these plants belong to the Apiaceae family. So it's possible that the similarities shared between these species belonging to the same family led them to have statistically insignificant differences in OTU composition, or very similar OTU composition. I wanted to take this idea and look at the other plant families that are found throughout my data. All of these plants that are highlighted here, acme, ascu, bade, ursp, and urla, all belong to the Asteraceae plant family. And you can see that all of the other red boxes in my data set belong to the Asteraceae family. So it seems that a plant family is driving some of the similarity that's found between some plant species. However, that wasn't universal. For ascu and acme, the p-value was 0.04, which is smaller than 0.05. Or which is, smaller than 0.05, indicating that there are differences between these plant species in their OTU composition. But notice that that value is actually pretty close to 0.05, especially compared to the other p-values, which are closer to 0.001 or 0.002. The same is true for ursp and urla, where the p-value is technically indicating that there is statistically significant differences in OTU composition, but they're not very strong differences because this p-value is not too much less than our alpha value of 0.05. However, there was one exception to my brilliant, wow, plants belonging to the same family all have very similar OTU compositions. Ocpho and Demi both belong to the Ranunculaceae plant family. However, they had a very, very small p-value, indicating that there are differences in their bacterial OTU compositions. We can examine those similarities and differences in OTU composition using an NMDS graph. In these kinds of graphs, data points that are close together have very similar bacterial OTU compositions, but data points that are large or that have large distances between them have very different bacterial OTU compositions. So if you look at my data set, you can see that species usually clump up into fairly distinct groups throughout my data set. These are just a couple of the species that I'll highlight here. However, in some parts of the graph, they're a little bit less distinct, where you see lots of overlap between plant species. So if you take these groups, and instead of looking at the species level, you look at the family level, you see that many of the differences in these clusters all sort of conglomerate into one large color. So let's take lout and loader, for example. Remember, these plants had very similar bacterial OTU compositions, and you can see in this graph of purples and blues, they all become one large cluster. Same thing for the asters. We had 5 different species present here that become one large group when you look at the family level. And interestingly, remember that Demi and Ocpho were 2 different species, or that they had very strong differences in bacterial OTU composition. You can sort of see that they're grouping into different sections here. But these 2 plants are very close to each other in ordination space and sort of become one large group when you look at the family level. Of course, there are a couple of other interesting factors of this graph that we still have to figure out. For example, we don't know why Luli is so close to Lout and Loter. Also, our Castanea parasite is sort of not as distinct as the other plant species are, where we have several species that you can sort of tell that there are groups clustering here, but they are quite close to one another. Also, there are a few outliers that may actually be representative of these species, but in different that something might be going on with these samples that makes them different from other plant species. So, for example, this was a Caprifoliaceae plant that ideally would have been clustered with the other ones, and this was an Asteraceae plant that was a little bit further out than all of its other plant samples. So, in conclusion, there are differences in bacterial OTU composition between plant species, which is amazing. And for plant species that don't have differences in bacterial OTU composition, they usually all belong to the same plant family. So plant species and plant family clearly has something to do with the bacterial OTU composition within plant stems. Next, I want to look at my second question. Differences in bacterial OTU composition between sampling locations. So remember, my two sampling locations were Smith Prairie and Glacial Heritage Preserve. These sites are separated by a distance of 95 miles, and Smith Prairie is cut off from the rest of mainland Washington. Since it's an island, it's cut off by the Puget Sound. So I was interested to know if these factors were significant enough to cause a difference in bacterial OTU composition derived from the same plant species. 4 of my 16 plant species were shared between Glacial Heritage Preserve and Smith Prairie: acme, farrow, urla, and kale. You can see that we're working with fairly large sample sizes, so that was good. But I could have spread out my sample collection between Glacial Heritage Preserve and Smith Prairie a little bit more evenly. It was easier to get to Glacial Heritage Preserve, so I sampled this site more often than I sampled Smith Prairie. Using my Permanova again, I found that acme was the only plant that had statistically significant differences in OTU composition between Glacial Heritage Preserve and Smith Prairie. This was a p-value of 0.04, so that's quite a strong p-value. You can also see in an NMDS graph, remember that points close to one another are similar and plants far apart are different. We have our Smith Prairie plants clustered in a group that's a lot smaller than the Glacial Heritage Preserve group. These groups aren't super distinct, but it's interesting to note how they tend to cluster away from the other Glacial Heritage Preserve plots or samples. Next, we have our urla and kale. Our p-values were 0.07 and 0.09, which indicate that there are no differences in bacterial OTU composition between plants collected at Glacial Heritage Preserve and Smith Prairie. However, these p-values are very close to 0.05, which indicates that perhaps with a few more samples, it could strengthen this test so that we could actually see if there are true differences between these sites. For farrow, however, the p-value is very large, so there really is no pattern of bacterial OTU composition and site location. You can tell that the Glacial Heritage Preserve sites and the Smith Prairie sites are all intermixed here. So in conclusion, there are differences in sampling location for acme, but for none of the other 3 species that I tested. Therefore, my results are not consistent enough to say if there is truly an effect of sampling location on bacterial OTUs collected from the same plant species. Next, I wanted to look at my question 1C, differences in bacterial OTU composition between site disturbance regime treatments. So remember back to that graph I showed you earlier, or that map. Glacial Heritage Preserve had 5 different treatments applied to research plots. We had our annual early burn treatment, our annual late burn treatment. So early burn and late burn basically mean that they were burned either early in summer or late in summer. Our triannual early burn, so annual meaning burned once a year, triannual means burning once every 3 years, not 3 times a year. And finally, our mowed annually plot, which contrasts to plots that were burned. It was quite difficult to collect enough plant samples across all 5 different treatments to amount for a test, a statistical test between plant species. So only kale, sear, and lout were chosen for use in this test. So I was essentially testing 3 different variables: seasonality, so differences in bacterial OTU composition between samples that were in plots that were either burned early in the summer or late in the summer; frequency, so plots that were either burned once a year or plots that were burned once every 3 years; and treatment type, so differences in plant OT or in bacterial OTUs from plants that were collected in sites that were either burned or mowed. 3 of the 16 plant species had a large enough sample size to test. So we can see that we have a fairly large sample size. It could have been larger had I collected more plants from these plots. And I wanted to collect enough samples so that we had an evenness across treatments. So you can tell that some plots didn't have quite as many samples represented from these plots as others, where the triannual early burn had many samples and the annual late burn perhaps didn't have as many samples. And that was just because it's difficult to control what plants I was sampling from these areas, since it's growing in a natural open prairie. So after performing another permanent test, I found that only sear had a significant difference in disturbance regime, where our p-value was 0.014. So in this NMDS, I had 2 overlays. One is shape, which is the date of our last treatment, because these plots were sometimes not treated on schedule due to fire season or difficulty in asking the prairie managers to come and burn those sites. But our colors refer to our disturbance regimes. So after performing a pairwise test, I found that there were differences in the bacterial OTU composition of plants taken from triannual early burn sites and plants taken from triannual late burn sites. So that goes to show that only differences were calculated for sear, and differences were only found between triannual early burn and triannual late burn sites. That might indicate that there is an effective seasonality for sear alone, but overall, there's not enough evidence to say that there is a difference between disturbance regime treatments. Next, I want to talk about the second part of my thesis, chapter 2, question 2, effect of parasitism. So, remember back to when I was describing my plant trios. We have our parasitic Castanea levisecta plant, which is theoretically parasitizing our host plant. We couldn't confirm parasitism in the field, but we assumed parasitism based on its proximity to our host plant. We also collected non-host plants that were at least 2 feet apart from our host plant and our parasite, just to make sure that this non-host was not being parasitized. However, we wanted to collect it from a nearby plot to make sure that microclimate wasn't affecting the bacterial OTU composition of these plant stems. So I took these non-hosts from neighboring plots, which essentially means that the plots were either immediately next to or diagonal from the host and plant plot that they were taken from. So, for example, if we take plot number 19, if I had taken my host and my parasite from this plot, the non-host plant could have been taken from plots that were diagonal to this plant, or plots that were immediately adjacent to this plot. So, once I had grouped my parasite plants into trios, I calculated the difference in bacterial OTU composition between plants, between the parasite and the host, to see how different their bacterial OTU compositions were, and between the parasite and the non-host to test and see how similar their bacterial OTU compositions were. Ideally, if the Castanea and its host plant were sharing bacteria, you would think that their bacterial OTU compositions would be more similar than the bacterial OTU composition of a parasite and a non-parasitized plant. So the differences between these plants would be small, and the difference between these plants would be large. I collected enough trios to test 5 different plant species: Bade, Cacu, Urla, Farrow, and Lout. The sample size for this test was fairly small, but thankfully paired t-tests are somewhat less sensitive to sample size than per MANOVA. So I performed my paired t-test, which gives us p-values, just like the per MANOVA test. And I found that there were significant differences in similarity of OTU composition between hosts and parasites and non-hosts and parasites. So for these plants, the host and the parasite were more similar to each other than the non-host and the parasite were to each other. My other 2 plants, Bade and Farrow, were also somewhat close to our p-value of 0.05. So although we didn't calculate, or we didn't end up with a statistically significant result, with a larger sample size, it could be possible to strengthen this test to truly examine if there is an effective parasitism going on here. For Cacu, though, there was certainly not an effective parasitism, as our p-value was quite large. So I wanted to examine this in a figure, where we have our host and parasite difference on this axis, and our non-host and parasite difference on this axis, where if hosts and parasites were similar to one another in their bacterial composition, we would see data points falling in this part of the graph. However, if non-hosts and parasites had a more similar bacterial composition than hosts and parasites, they would fall into this part. So I theorized that if plants were sharing bacteria via parasitism, we would see many data points falling above the graph. So this is my actual data. First, you'll notice that many data points fall fairly close to the 1-by-1 line. Points that fall along this line show that the bacterial OTU compositions of hosts and parasites are not smaller or larger than differences in host plant or non-host plant and parasites. Interestingly, for many of my other trios, we find that the sample data points fall above the line, which indicates that there could be an effective parasitism happening for these points. So if we look at Urla and Lout, my 2 plants that had a statistically significant difference in the similarity between hosts, plants, and parasites, and non-hosts and parasites, we see that for Urla, 2 of the trios fall along this 1-by-1 line, indicating that there isn't an effective parasitism. But for the other 4 trios, they fall quite far off the line, which indicates that there is a fairly strong effect of parasitism that might be driving similarities between the hosts and the parasite bacterial OTU compositions. For Lout, notice that all of the data points fall off the 1-by-1 line, but they don't fall off quite as far as the Urla points. So that was just interesting to note. Overall, only Lout and Urla showed an effect of parasitism on OTU composition. None of the other plants had an effective parasitism on OTU composition, although 2 of my other plant species had a fairly small p-value. Overall, though, I can't say that there is enough evidence to say that parasitism could be driving similarities in bacterial OTU composition of these plant species. So, just to summarize, my chapter 1, does the bacterial OTU composition of samples differ between plant species? Yes. we did observe that plant species tend to have very different bacterial OTU compositions. And for plant species that had similar OTU compositions, those plants belonged to the same plant family. However, I didn't find that there was a difference in bacterial OTU composition of plants collected from Glacial Heritage Preserve and Smith Prairie. So there was no effect of sampling location on bacterial OTU composition. Next, I did not find an effect of disturbance regime treatments on bacterial OTU composition, at least at Glacial Heritage Preserve. Finally, Chapter 2. Are the bacterial OTU compositions of hosts and parasites more similar than for non-hosts and parasites? No, I did not find that the hosts and the parasites had a more similar bacterial OTU composition than non-hosts and parasites. So now that we have this research, we're starting to think of other ways that we can use this data. If you enjoyed this presentation, I would highly recommend that you go check out the Earth Microbiome Project. They have some information on their research and their goals online. And beyond that, our lab group is interested in studying the indicator species analysis on this data. So we're interested in finding out if some bacteria only associate with certain plant species or plant families. Additionally, we had planned an experiment to see if bacteria could travel between kale and its host in a controlled lab experiment, where essentially we would take bacteria from a host plant, take it out of the plant, make it glow, stick it back into the host plant, and then watch and see if we could see that bacteria travel between the two plants. So I would like to open up the presentation for questions. Thank you very much for all joining in on my presentation.
Thank you very much, Tori. So if you have questions for Tori, we've got a good 10 minutes for those. And you may either unmute yourself and speak up, or you may put them in the chat window and I will then communicate them to Tori.
And it's very quiet. Don't be nervous. I promise I'm not nervous.
All right, we got some questions coming here.
I have a question. Oh, can I speak?
Yep, go ahead, Dave.
Yeah. Hi, yeah, this is Dave Wilderman. I'm just curious, in one of your earlier slides referenced something like close to 6,000 or 7,000 different OTUs amongst all your samples. I guess I'm curious if there's any other studies out there that would provide some context for that number.
Right. Like
yeah.
other studies to see just how many different bacterial OTUs can be found within plant stems?
Correct, yeah.
Right. Well, there have been some other similar studies out there that look at the different kinds of bacteria that can be found in plants. I know of one just off the top of my head that also looked at the bacteria transfer between parasitic plants and host plants. And research like this commonly cites how many different bacterial OTUs are found in their data. It's probably worth noting that the 7,365 different bacterial OTUs that I had calculated were the total different number of OTUs across 292 plant samples. So the more plant samples you have, the higher likelihood of different bacteria you're going to find in your study. I also looked at 16 different plant species, so the larger breadth of different plant species that you have likely would also increase the number of different kinds of bacteria you might encounter. And does that help?
Yeah, okay. Yeah. I was just curious if there were any similar, and maybe there aren't, similar studies out there that addressing similar numbers of plant species or samples that would provide some comparison, but maybe there aren't.
Right.
So,
Well, as far as we know, this is the only survey that's been done on these 16 different prairie plant species. So that's sort of exciting, novel research on our end.
yeah, that's great.
Tori,
That's great. It's interesting. Thank you.
Tori, could you maybe speak to other studies that have, like, how many OTUs per sample have other studies found on average? Do you have an idea of how yours compare that way?
Yeah, I actually don't know that information off the top of my head. Studies don't usually talk about that in their data collection because probably due to like the brevity that they have to include in their results section. So I wouldn't actually know how well these numbers compare to other studies.
Okay. David Zuckerman had a question. What determines the difference between high quality and low quality DNA sequences?
Right. So that is something that's usually determined by the Illumina software that performs the sequencing itself. So sometimes there can be reads that have low quality base pairs. So a base pair is an A, T, G, or C. Sometimes the identity of that base pair isn't all that high in quality. And that can be due to a couple of different factors in how many other different particles are present in the sort of little set that you have while you're sequencing. So that's usually something that's investigated, or like the quality of that base pair is calculated using the machine that is performing the sequencing. So each base pair is assigned its own quality score, and we usually only want reads that are of high enough quality where we can be fairly certain that we are dealing with a string of that identity. So an A isn't just, an A has a fairly high quality score, so we're fairly certain that it is an A. But if an A has a fairly low quality score, the machine wasn't exactly certain if that nucleotide was an A or not.
All right, and then a question from Elaine. The bacteria seem to be identified in very broad taxonomic groups, phylum level. Is it possible to look at them at a finer level?
That is a wonderful question. Yeah. So in my analysis, I originally wanted to look at lower taxonomic levels of bacteria. So it was possible for some of my bacterial OTUs to look at their class and their order, and sometimes their genus and species level. But for only a few of my bacterial reads could I identify them down to the species or genus level. That's just because we have to know so much about the reads that we're dealing with, and the database has to have that information stocked in it. So yeah, the phyla level is one of the higher taxonomic levels that we could look at. And it was also easier to look at that taxonomic level because there weren't so many different kinds. Like as soon as you start to look at like the class or the order level, instead of having 26 different bacteria, now all of a sudden you're looking at 100 or 150 different plant or different bacterial taxa.
But Tori, in your analyses, just to clarify, you were using the OTUs in the analysis. So that is the smallest possible scale for the analysis itself. Right? The figure that you were showing the phyla on is just a summary, but that's not the, you didn't analyze it at the phylum level. Right?
Exactly. Yeah. My analysis occurred at was performed on the OTUs itself. So the presence of the OTU and the abundance of the OTU, not necessarily the presence of phyla or class or order. That's sort of why I introduced that section before I began looking at my research questions, because those are two different ways of looking at what OTUs I have. Good question.
All right, and then a question from Karen. Great presentation. Could you say a bit more about the ecological or organismic role played by bacteria? In other words, how do we think of them functionally in ecosystems or organisms?
Yeah, that would refer to the large part of my introduction in my early parts of my thesis, if you would really like to get into that information. So essentially, bacteria serve many different functions in the natural environment. Some bacteria we refer to are endophytes, which are bacteria that positively influence plant growth or neutrally influence plant growth. So these bacteria are able to interact with their plants by providing nutrients in exchange for energy. So those bacteria are usually thought of as beneficial bacteria in our system. We also have pathogenic bacteria, which can have maleffects on the plants that they inhabit. So in my study, we don't know the identity of my bacterial OTUs at the level of, are these bacteria beneficial, neutral, or pathogenic? So the bacteria that I have in my sample could have served many, many different kinds of ecological function. And just separating bacteria based on beneficial, neutral, or pathogenic effects on the plant hosts that they have is just one way of looking at how these bacteria interact with their systems.
All right, and then one more question here from Matt, and if others have got questions, you're welcome to, again, post them in the chat or to speak up after this. Matt says, thanks for an interesting presentation. I'm a bit concerned that the cyanobacteria in the data reflect unintended PCR amplification of DNA from chloroplasts. Did you try to prevent amplification of plastid DNA in your library preparation protocol?
Fantastic question. So as we were extracting this DNA and sent them out to the University of Minnesota Genomics Center, we asked them to include PNA blockers that would exclude certain kinds of bacterial strings. So we included blockers that would block mitochondria and that would block chloroplast. And after having retrieved my reads from the University of Minnesota Genomics Center and trimming these reads and identifying them in the software that I had, I took a cursory glance through the different kinds of bacteria that I had, and I actually had a fairly low representative portion of bacteria that could have been identified as chloroplast.
Other questions for Tori? We have time for one or two more. So Zoom calls are really nice for being able to have everybody participate remotely. One thing they don't do very well is allowing all of us to applaud simultaneously. Please join me in thanking Tori for her participation. And if you've got additional questions for her, you're welcome to reach out to her. So Tori and Sharon and I will stay online here. And thank you all for your participation.
Yeah, thank you all very much for coming to hear me talk about my research.
There's a number of congratulations in the chat window, Tori, for you to check out.
I see that. Thank you so very much, guys. I always enjoy talking about my research, so if you want to reach out to me after this presentation, I would be more than happy to hear from you guys. It was great to see so many friends and family in the crowd too.